Started. Welcome to the first faculty seminar series of 2026. Pleasure to be here. Thank you for making time for this. For those of you that don't know me, my name is Rob Duncan, and I am happy to be here, and my pleasure to introduce Dr. Carla Taylor. So just a quick intro on Dr. Taylor. Is a professor in food and human nutritional sciences and the principal investigator of the Metabolic Nutrition Laboratory at CCARM, and was the leader of CCARM from 2016 to 2022. Dr. Taylor received her bachelor's from the U of M and PhD from the University of Guelph, and did some postdocs at University of Michigan, Ann Arbor, and University of Washington in Seattle. So Dr. Taylor, her program mainly focuses on dietary components such as lipids, proteins, and bioactive compounds from plant, marine, and animal sources that are beneficial for the prevention of and management of metabolic diseases, particularly obesity, fatty liver disease, and cardiovascular disease. And her research investigates potential mechanisms linking cellular, interorgan, and whole body metabolism. And Dr. Taylor in 2025 received the Earl Willard McHenry Award in recognition of distinguished service in nutrition from the Canadian Nutrition Society. So it's my pleasure to welcome Dr. Taylor. Please join me in welcoming.
Good afternoon, everyone. I'm honored to be asked to give one of the presentations in the faculty seminar series this winter. And thank you all for coming out this afternoon. So I want to share some of the things that I've been doing, which in kind of the broader sense are about linking agri-foods with health. And I'm going to be focusing on some of our findings around omega-3 fats and dietary proteins for management of metabolic disease. And so this afternoon, I've kind of chosen certain things to kind of highlight that I think are of interest to me, hopefully to you as well. For some things, if you wish to know some more details about it, you know where to find me afterwards. So this is going to be hopefully some interesting information. But I thought before we start talking about what I've been doing, this whole concept of linking agri-foods for health, right, is certainly not a new one. And so I'm wondering in this room, who knows what Dr. Bruce McDonald and Dr. Vivian Bruce are known for their contribution to canola oil research? Anyone? Any guesses?
Fatty acid analysis?
Sort of, yes. Going a little. So yeah, so they were doing some of the very first human studies with canola oil, and so this is something that Dr. McDonald wrote for the story of canola oil, that the initial studies were completed in 1972 to 1973 in response to the decision by Canada to replace the entire rapeseed crop with canola by 1975, right? So all the plant breeding had been happening, oil was being produced, but the question, right, safe for humans, is it going to have some positive biological effects? And so they were conducting those initial studies. And along with that, in the Department of Foods and Nutrition, Professor Marion Vasey-Genzer and Dr. Michael Eskin were conducting studies related to cooking, well, they called it cooking properties, the way it's written up, but shelf life and deep fry stability of canola oil. And then, of course, various other things had to fall in place as well in terms of the production and distribution, so that canola oil began showing up in the human diet. And so I thought it might be of interest just to quickly look at one of the first papers they published. And so you can kind of see the study design here at the bottom. It was a crossover design. So they were all male participants here, students probably at the University of Manitoba campus that were in these first studies. It was a crossover study. They started with a wash-in mixed fat diet, canola oil diet, switched over to sunflower oil diet, and back to the mixed fat diet. And so the outcomes that were being focused on were the lowering of cholesterol and also what's called this antithrombotic effect, which is looking at how quickly blood clots. Okay, so it's actually an advantage if it takes a little bit longer for our blood to clot. I mean, we still need our blood to clot, but a slower time to clot decreases the risk for heart attacks and strokes, right? And that's the basis of medications that some people take. And so in that bleeding time, we can see with the canola oil diet, after just 18 days, it significantly changed, took a little bit longer for the blood to clot, which is an advantage. And interestingly, these days, with some of the things that are out there on social media and elsewhere about different types of fats and so forth, I find it interesting that the mixed fat diet did contain beef tallow, as an example, lard, corn oil, butter, vegetable shortening. That's what it was being compared to. And you can see here the advantage of bleeding time, anyhow, and also lowering of cholesterol with having the different types of fatty acids in the diet, right? And here I'm kind of relating to some of the stuff that's out on social media right now about the Human Health Secretary in the US, right, making some, I'll say, claims about going back to beef tallow, for example. And so I think, well, maybe he should take a look at this paper as an example of some actual scientific data on the topic. Okay. And so these diets also different in the composition of the types of fatty acids that were there. And this is all kind of part of the human nutrition story around how the amount and type of N6 and N3 fatty acids can change what the biological responses are in the body, and for health, but also for helping to protect us against certain diseases. So you noticed in my title, I had metabolic disease. So kind of a precursor to that is what's known now as metabolic syndrome. So metabolic syndrome is kind of these clustering of factors to do with high blood pressure, increases in the blood sugars, changes in some of the blood lipids. But it's just kind of an early stage. It's kind of a pre-stage to cardiovascular disease or a pre-diabetes stage, as an example. And then as these develop to the metabolic diseases, we can see how they affect different organ systems and different diseases. So cardiovascular disease, diabetes, liver disease on the far right, but you can also see that dementia and cancer are on this list as well. And so my interest in the research that I'm going to be talking about today is going to focus on obesity and adipose tissue, steatosis in the liver, and cardiovascular disease and the health of blood vessels. And I should also say, when I say we or our, this research represents a whole team, right? Because to me, research is multidisciplinary, it's a team approach, and it's been great to have so many different trainees, employees, and collaborators contributing to all of this work. Okay. So I'm going to say a little bit more about this fatty liver disease, because this is something which is emerging now, and it has a title now, Metabolic Dysfunction Associated Steatotic Liver Disease. So you'll understand why I'm just going to abbreviate and say fatty liver disease when I'm talking about it this afternoon. The former name was non-alcoholic fatty liver disease. So this is when there's an increased amount of fat that builds up in the liver, and it's not due to alcohol consumption. Okay? So the metabolism has changed, and there's a progression here from simple steatosis. So this is by definition when there's greater than 5% the weight of the liver is composed of fat, and that level can keep going up further. Then it can progress to MASH, which is when there's an inflammation involved. Cirrhosis. Cirrhosis gets to end-stage treatment, and the only treatment at that stage is a liver transplant. So this is because why it's so important to recognize early on when the early stages are happening, and how we can use, say, diet and lifestyle as one of the approaches to manage these. So fatty liver disease is associated with obesity, diabetes, and cardiovascular disease. It's estimated that maybe 15 to 30% of the population has fatty liver disease. And in most cases, people don't know because the diagnosis depends on having some sort of imaging technique. Okay? So there isn't a reliable method of, say, doing a blood test to determine whether or not someone has fatty liver disease. So if, say, for some other reason, someone has an imaging for diagnosis of something else, it might be recognized, oh, they also have fatty liver disease. The most effective treatment is weight loss, which we also know is very challenging for most people. And most recently, the FDA has approved the first drug, which is specifically targeted to managing this disease. Okay? But right now, the treatment is really around weight loss. And our question has been about what sort of dietary constituents can help manage this more effectively. So whether we can slow the progression or whether we can actually help treat the actual disease. And so this is where I'm going to bring in the omega-3 fatty acids. Okay? And I think in this audience, probably everyone is familiar with this nomenclature. So, and the sources being for ALA, the first one here in the series, alpha-linoleic acid, 18 carbons, 3 double bonds. So plant-based sources with flax and canola being sources of ALA. In our body, ALA can be converted to EPA and DHA, so the longer chain versions of these, which EPA and DHA can be found preformed in sources, deep ocean fish, so salmon, for example. In the area and also within the research we've been doing, we've had specific questions about, is there something specific that ALA can do without being converted to those longer chain EPA and DHA, or the fact that ALA is an essential fatty acid, can we essentially achieve all of its functions via its conversion to EPA and DHA? Okay, so those are some of the questions in the area and some of the things that we've been addressing. And so if we apply this to omega-3 fatty acids and which one or ones might be effective for decreasing hepatic steatosis, one of the studies that we did used this genetic model of obesity. So you can see a picture there of the FFA Zucker rat. And this was a 8-week study where we had various experimental groups. We had a baseline group so we could compare how they started to after 8 weeks on the different diets. there's different advantages and disadvantages of different models. So with the genetic model of obesity, the disadvantage is that it's a single gene mutation that's creating that obesity. So that doesn't necessarily reflect obesity in humans and saying being a multi-gene effect. But the advantage is that we can use a relatively low-fat diet and study what is happening in the context of obesity and the changes that we're making in our diet. An alternative model is to use diet-induced obesity in rodents. And so with that, you're using a very high-fat diet. So it might be anywhere from 45 to 60% of calories from fat. So then that also changes the context of how you're approaching the research as well. So for this particular study, we're using the genetic model of obesity, 10% by weight dietary fat, with equivalent amounts of saturated, monounsaturated, and polyunsaturated fatty acids in all of these diets. And the reason I'm making this point is if you look in the literature at many studies that are changing the source of fat that they're looking at, it's like, yes, they're changing the source of what they're looking at, but they've also changed the percent of, say, saturated fat or monounsaturated fat that's there. And then that's also having effects on what's happening and will be confounding the design. So in this case, we are only changing the type of polyunsaturated fat that's in these diets. So the ALA diet had a mixture of flaxseed oil and canola oil in it. For the EPA and DHA diets, we purchased purified sources because we wanted to study these individually. And then we compared this to diets that had linoleic acid, so an omega-6 fatty acid, as the primary source of the polyunsaturated fats. And what we found when we looked at the livers is that DHA was the most effective for preventing the progression of the hepatic steatosis. So we can measure the actual amount of fat that is in the liver, and we can see that the DHA group has a similar amount of fat in the liver as the baseline group. So that's how we can say that we prevented the progression of the disease. The EPA group was also not statistically different, but we can see it's kind of intermediate in the effect between the DHA and the ALA diet. As part of doing these studies, we section the tissue so that we can look at it under the microscope. And on the far right there, we're using a stain called oil red O. So that stains the lipid droplets that are in the liver. Okay? So in the liver, when it's accumulating fat, it's fat droplets that are actually surrounded by a phospholipid layer that is storing the fat. And as we're, say, after a meal, we're storing more fat in those lipid droplets, and then when we're in the fasting state, the triglycerides are being released so that they can be used for energy or go to other sites in the body. Okay, so it's a very dynamic process, and these lipid droplets are not all the same size. Okay, so there's actually a distribution of sizes. And so the reason I got the green star on here is to remind me to say that as part of this seminar series, one of the themes is around digital agriculture. And so I kind of had to think, okay, what does that mean in terms of what I do with nutrition? And so one of the definitions, right, is around how we use technology to collect data, analyze it, and so forth. So one of the things that we're doing here with these liver sections is we're using a software to be able to, the software actually identifies the lipid droplets, counts the number of ones in a particular area, can also determine what the size of those lipid droplets are. So we can get either the overall area of the lipid droplets, which is in panel B, or we can start looking at what the distribution is of different lipid droplet sizes. And so the DHA diet, which is reducing the amount of fat in the liver, has one of the fewest of the very large droplets, and it has a lot more of the smallest lipid droplets. Okay, so that's kind of some of the dynamics that are happening with the lipid droplets. Since we also in these studies look at more in terms of like pathways that are involved, I'm just showing here fatty acid synthase. So we're looking at what is the production of lipid in the liver. And it was the DHA group which has the lowest level of this fatty acid synthase, right? So that kind of makes sense that if we've got less fat present in the liver, there's less synthesis happening in this case. And that is part of the explanation in this particular study. So in the introduction, it was mentioned, right, I like understanding kind of what's happening at a cellular level, and then how does that relate to the whole organ, to the metabolism of the whole organism, right? So if we start looking at what's happening with blood glucose and blood insulin, we're starting to get more into that concept of what's happening between different organs and what's happening with, say, insulin sensitivity or insulin resistance. And so in this study, there was no difference in the fasting glucose, but fasting insulin was lower in the DHA group. And that kind of fits that if there is less lipid being stored in a tissue, it becomes more insulin sensitive. There's a calculation that can be done, this HOMA-IR, which is a measure of insulin sensitivity, insulin resistance. So here with it being lowered actually by the LA group, the N6 group. Okay? And so one of the things we're going to start seeing with the results I'm presenting is there's no one pattern sometimes of only a particular fatty acid is explaining all the changes. There's kind of these complementary things going on. Okay? And then in the last panel, this is again looking at what's going on in pathways in the tissue. And on this slide, I haven't yet talked about EPA, but yet EPA is the group where there's a protein that's lower, and this suggests that there's less gluconeogenesis going on. Okay? So the previous slide, I gave something to say DHA is affecting the fatty acid synthesis. On this slide, it's the EPA is kind of focusing more on the carbohydrate pathways of the metabolism. Okay, so this is work that's done in animal models. And if we kind of want to look at, okay, so what is the translation to humans of studies like this and work that other people are doing, is that more recently now there are various reviews and meta-analysis coming out about using N3 fatty acids as a way to manage fatty liver disease in people. Okay? And so there are more recently studies happening in humans to look at this as a particular approach. Okay. Okay. I'm also interested in what's happening in adipose tissue at the same time, right? Because in our body, there's all this communication and connection between different organs. And so adipose tissue is actually a very active metabolic tissue. Okay, so just like I mentioned in the liver, how the lipid droplets are storing and then releasing lipids continually, the same thing is happening in our adipose tissue. And even though sometimes we might think that adipose tissue is rather static, there's labeling studies that have been done to show that the adipocytes or the fat cells in humans completely turn over in 7 years. Right? So it's a very dynamic tissue. It also plays an important role in inflammation, Part of the reason is that we have immune cells that infiltrate into the tissue, and it's both the immune cells and the fat cells themselves that can produce molecules that are either pro-inflammatory or anti-inflammatory. Okay? And both types of molecules are being produced. It's just kind of what is the balance of it overall. And so when we look at sections of adipose tissue from lean versus obese animals, we can see differences in the size of the adipocytes, or the fat cells, so smaller in the lean, larger in the obese. This was a study done in older FAFA rats, and in that study we just gave flax oil as the N3 source, and we did see a beneficial effect in that particular study. And also in this study we saw that there was less infiltration of the immune cells. Okay? So this is kind of part of the story around omega-3 fatty acids being anti-inflammatory. And when we actually measure some of these markers, we can show that there's a decrease in pro-inflammatory markers. But what was really interesting about this study is that we can see these changes in the adipose tissue despite the amount of adipose tissue staying the same. So there's no weight loss and there's no change in the amount of adipose tissue. But yet what the animals were being fed, in this case omega-3 fatty acid, was in a sense making this a healthier adipose tissue. And I know that might seem kind of contradictory, but I think it's really important, right? Because I think sometimes in terms of thinking of this in terms of human nutrition, sometimes there's a lot of focus on what's actually happening with weight itself, but we also need to be focusing on what are the characteristics of the tissue and how is our diet and lifestyle improving its function. Okay. In this study that I was showing you with the liver and the different types of omega-3, we also looked at the adipocyte size in those animals, and we found that both EPA and DHA could reduce adipocyte size in those animals. We also looked at the tissue in terms of markers of immune cells. And so here in the bottom panel, this is a marker of macrophages, so a type of immune cell that can go into adipose tissue. And so in this case, it was especially lowered by EPA. There was also some lowering by both ALA and DHA compared to the NSITS diet. There was also a lowering of T cells, so another type of immune cell that can enter into the adipose tissue. And in this case, it was just in the DHA group that happened. And then if we were to look at some of the different markers, different things are happening in terms of which omega-3. And the reason I'm presenting this, and it might seem, oh, this is kind of getting really complicated, is I want to get the message out there that yes, it is kind of complicated when we're talking about pro-inflammatory and anti-inflammatory. And I think some of the messages that are getting out there in the media right now are just a huge oversimplification, right? Because there's this kind of statement out there right now, oh, omega-6 is pro-inflammatory. But I can dig into some of my data and say, no, we know we need omega-6 because linoleic acid is also one of our essential fatty acids. And if we look at different results, we have to look at the balance of what's happening, okay? And so if nothing else, that's a message, kind of a take-home message. And so also depending on which omega-3, we're seeing different effects happening on inflammation and on immune cells. Okay, so that was kind of a part around liver and adipose tissue. So now I'm going to move into a little section that's got to do more with cardiovascular disease. And the little picture that I've got up here is a blood vessel, and this is kind of part of the process that happens when we develop atherosclerosis, right? So when we have plaque, we get lipid deposits happening in our blood vessels. And so on the previous slide, right, when I was talking about adipose tissue, I was talking about macrophages. You can see here we have another type of immune cell, which is called monocytes. And so these monocytes are in our blood. And what happens in atherosclerosis is that a monocyte that's in the blood crosses through here into the blood vessel wall, and it becomes a macrophage in here. And then these macrophages are, in a sense, trying to clean up extra lipids. So that's how they become these foam cells. So one of the questions that we've had around the omega-3 fatty acids is whether they can affect this process of how monocytes bind to the blood vessel wall and then go into the blood vessel. And so this is an example of doing something in cell culture to kind of mimic this. So we can take a cell line, this THP1, so they're a monocyte type cell. We can label them with fluorescence inside, and then in the culture dish have endothelial cells growing. So those are the types of cells that are lining our blood vessel wall. And then we can measure via the fluorescence what in the environment is either encouraging those cells to have that adhesion or not. And what we found in this study was that DHA was actually reducing the adhesion. Okay, so that's a good thing, because if we're not getting as many monocytes adhering to our blood vessel wall, then that's going to reduce the chances of those monocytes getting into the blood vessel wall and contributing to the plaque. Okay, so that's a way that we can look at some of these mechanisms from a cell culture point of view. And then also we used ALA and DHA treatment of these immune cells to do a whole transcriptome analysis so that we could look at what are all the different genes which are being affected here, right? And probably in each of your disciplines, right, there's ways in which transcriptome analysis is being used as well. So this is another way, right, from kind of a digital technology type way that we can start looking at, okay, what is ALA affecting versus DHA? So you see we came up with, that was the list of top 10 upregulated genes. So some are the same, some of them aren't. And then in doing this type of work, right, it's good to go on and do validation. And so I'm just going to highlight a couple of things that we found. They're published in this particular paper. So we could treat additional cells and validate changes that were happening. And this particular one is of interest because HMG-CoA reductase is the rate-limiting enzyme in our body for cholesterol synthesis. Okay? And this is the drug that statins target, that statins are inhibiting. And so this is why it was really interesting that it came up in our list of kind of top hits from that transcriptome analysis. So we could confirm with our cultured cells that both ALA and DHA are treatment of those cells is reducing the expression of that gene. Because we had been doing clinical trials, we had some stored samples of immune cells from people who had been consuming either ALA supplements or DHA supplements for 28 days. So we could verify in human cells that people who had consumed either ALA or DHA, that there was a reduction in this particular HMG-CoA reductase. And it's nice to find things happening at, say, an mRNA level, but the question is always, does it pan out in terms of an actual function? And we took these cells and treated them with DHA, ALA, a statin, and we found that the cholesterol content was reduced. Okay, so this is kind of nicely fitting together that these omega-3 fatty acids are having an effect on our cholesterol pathway. And also you'll see in here we included an N6 fatty acid and LAs as a control, so that we know this is specific to these omega-3 fatty acids. So a PhD student in the lab right now, her whole project is kind of a takeoff of this angle and looking more at cholesterol-related pathways and related to omega-3 fatty acids. The other thing I'll mention from that transcriptome analysis, it was interesting that ALA and DHA tended to do the same thing, similar things, when they were down-regulating pathways related to fatty acid and cholesterol metabolism. Okay, so they're kind of acting similar in that way. And then on the flip side, when there were pathways that are being regulated, and they are being upregulated by ALA, it was kind of only ALA was doing it, not DHA. So this is where we could kind of see some differentiation in what was happening. And so this EGR1 is a gene that's involved in multiple cellular processes. It's related to infection. So again, we could see the increase with ALA treatment in cells, ALA supplementation in people, but not with DHA supplementation. This time when we wanted to look at a functional assay, we don't have something which is really specific to this gene. We tried something related to immune function, and in this case, it kind of doesn't follow through, but we also know it's kind of not really specific. So, so that's kind of also a challenge sometimes in being able to confirm what's happening. Okay. So I haven't been talking, well, I've kind of been alluding a little bit to, you know, how are some of the mechanisms of how omega-3 fatty acids work. This slide kind of summarizes kind of three kind of branches in terms of how they might be affecting membranes and processes related to membranes, being substrates for signaling molecules, or actually working via extracellular or nuclear receptors in the body, right? So for those of you who are interested in that aspect, what I'm going to focus on next is just an example of how we've been looking at the middle part, how fatty acids and omega-3 fatty acids are substrates for fatty acid metabolites known as the oxylipins. Okay, and so this work is done in collaboration with Harold Akama, right? And so this is when having collaborators in your department, right, who are experts in your field, and what you can do collaboratively is really important. Okay? So various N6 and N3 PUFAs can be used to make oxylipins through various pathways. And one of the days that our group was kind of sitting around the table trying to figure out where we wanted to head next with some of our work, and especially thinking about sex differences, led to this particular study about where oxylipins, they somehow involved in explaining sex differences in cardiovascular disease, right? Because premenopausal women are protected from cardiovascular disease, but postmenopausal women have similar disease rates as men. Okay, so we kind of put our oxylipin focus on this. We had archived samples. We were able to do the plasma oxylipins. We had a whole set of clinical data. The samples that we used, we knew the blood vessel health of people because we work with this non-invasive equipment to know what the blood vessel health is. And I got the green star here because large amount of data here that got reduced down to this, our graphical abstract. It came down to that we identified 3 of these oxylipins. Okay, so that's, and the blue, those are just abbreviations for them. So premenopausal women had higher levels of those 3 oxylipins, and they had more elastic vessels. And again, because of their age. But postmenopausal women and older men had decreased levels of those oxylipins, and they had stiffer vessels based on our pulse wave analysis. Okay? And it's also interesting that these 3 oxylipins are all made by an enzyme 12-lipoxygenase. Okay? And they come from different fatty acids. So we're kind of trying to figure out that part now. And so I'm going to be talking about a clinical study that we're doing next, but actually Jordan, who's here today, a master's student, she's working on the 12-lipoxygenase part as part of her thesis. Okay? So when we started into this, I don't think we thought that it would necessarily come down to something, you know, kind of this neat in a way, but sometimes you never know what's going to happen. And this has led into further funding and a CHR project that we're doing right now to actually specifically recruit people and do the prospective study on this. So that's what we're doing right now. The 4 groups of individuals. We have our younger individuals already recruited, and I know Crystal's been very helpful helping to get the message out about our study, and so we've had a good response, and thank you to people in the faculty who have responded. We're still recruiting in the bottom 2 groups, and this is where we're now going through again and trying to link this fatty acids, omega-3 index, oxylipins, hormones, with dietary intake and arterial stiffness. And the follow-up of this is going to be that we'll do a fish oil supplementation study. We originally thought that we were going to focus only on people that have a low omega-3 status, but when we've got our results back from about the first 100 people, almost all of the people, I'd say about 80% or more of the people that have come into our baseline study have low levels of omega-3s. So, so even though we're not going to make that a requirement for that part of the study, that is the group that we'll be studying just by nature of what the typical diet is here. Okay? Okay, so that's kind of my part on omega-3 fatty acids, and the next part on the proteins is just going to take a few slides. So this is going back to the liver, steatosis. Remember what I said about this fatty liver disease. What can we do with dietary constituents? There are well, in this case, this is an expert consensus about what should we be doing about diet. And you can see there's some very general things there, healthy sources of protein. Even though it says primarily plant-based, there's not really data to support that. That's kind of a perception about that. There is some evidence that high-protein diets can reduce liver fat and improve blood glucose-related parameters in individuals with type 2 diabetes or fatty liver disease, but those studies are really just about high-protein diets. They're not much into which type of protein is going to be most effective. And so that was our question. And the high protein diets that we're doing in these studies are 35% of calories from protein. So that's at the upper end of what's recommended for humans. So this isn't something which is super high. And this study, we again used that Zucker rat model. And you can see the top part that's in blue. So we've got the lean on casein, and then we have normal protein level of casein and high protein casein. And those are a lot of lipid droplets. That's not a particularly good thing. Down here, soy protein, so it was better, and that's kind of what was expected. What we weren't expecting is that this diet, which was a mixture of animal and plant-based proteins, was really effective in, at least in these Zucker rats, for reducing the liver triglycerides. And so we could show that by measuring the fat in the liver. This study was initiated by Harold Akama from the kidney perspective, but at the same time they said, well, let's see what's going on in the liver and some of the other tissues. And so this had already been designed because Harold Akama and Jim House had done a study in pigs because they were interested in high protein diets in kidneys. And so this was kind of already predetermined, and we thought, well, we'll try it out in the rat model. And it had benefits. So then we realized, okay, protein source is important. And it took us a little while to get some more funding to follow through on this. And I'm going to do this as a reveal. So you can see column A is just pictures of the livers. B is an H&E stain. C is with the bright green, that's a fluorescent stain for lipid droplets. And then D is a stain for fibrosis, so where there's some red, and it might not show up too well in this. So you can see how the rats started out. Here's our casein diets again. Okay, and it's interesting, this is the standard protein that's used in rodent diets for, they call it the AIN 93 diet. And we've repeated, I know there's some other studies out there now showing that just casein by itself is not having a great effect. So then, what do you think these last three are? Because you can see the liver looks better, right? So next one is egg white protein. Right? So we had to come up with proteins that are available, right, as a protein isolate. So egg white protein. The next one is our mixture diet of egg white plus 2 plant proteins. So we did a combination of soy and pea protein. They're being kind of the most used sources right now. Right? So it's easy to buy soy as a ingredient for animal diets. Pea protein I bought off of Amazon, actually, because the companies that produce pea protein aren't exactly interested in having animal studies done with pea protein. So there's kind of a philosophical thing there. So just so everybody knows that that's how I got that particular pea protein. And then at the very bottom, we have the plant protein diet as well. So when we quantify what's happening in the liver, you can see here the last 3 groups. So all pretty similar, but there is a statistical difference here. When we improve what's lowering the fat in the liver, we improve this measure of insulin resistance. And if we're improving the insulin resistance, we also want to see what's happening in the pancreas. And so here where we have smaller pancreas islets, it means that the pancreas isn't being stressed as much either, right? So this is a multi-organ effect that's happening in a positive way. Okay. Of course, there's always something a little bit surprising, usually in most studies, and so this is the growth curve, and the two groups here that weigh more during those last weeks of the study are actually the plant group and the MITS group. Again, I don't know if it was a palatability issue with the diets or if there truly is a difference there. But there are a few studies in the literature, though, now showing that there can be a higher body weight with plant protein. So what that means, not exactly sure. And so again, we've been studying more the underlying reasons of why this is happening, and it actually happens with the egg white and our plant protein groups. There's a better export of lipid from the liver, which is helping to explain that lowering. And again, so a final conclusion, right, that amelioration of hepatic steatosis by high protein diets is dependent upon the protein source, and it does not require weight loss, right? And the fact that we did this in younger animals, I mean, that bodes well for, say youth that are growing but have this condition. Okay. So lastly, here's just a few summary points. Omega-3 fatty acids have distinct but also complementary roles for the management of metabolic disease, right? I think we need a combination of all of them, right? We can study from a basic science point of view what specifics different ones are doing, but at the end of the day, they're all contributing in positive ways. I've already mentioned about the hepatic steatosis, high protein source being important, how we can improve the health in the liver of the adipose tissue in obese states without weight loss or changes in organ weight. I've shown you some examples of how tools and technologies are advancing how we can collect and analyze data, and that linking Agri-Foods with health requires a multidisciplinary approach. Okay, so lastly, I'd just like to acknowledge everyone that's been involved, and all my collaborators, many more than are listed here, different facilities, all the funding, scholarships received by trainees. So that's what makes it all work, makes it fun. So thank you.
Dr. Taylor, really enjoyed that. That was educational for me, for sure. Any questions from the audience? Yeah,
thanks, Carla. I'm just interested, have you tried like mixture of protein and the omega-3 fatty acids to see whether there's synergistic effects between those two products?
Yeah, we haven't. We haven't designed a study to do that. But what's interesting is I look back over some of the work I've done because I started out doing zinc deficiency. We always used egg white as our protein source because it has less zinc present than, say, casein. And so the egg white as the protein source, I kind of carried over into some other studies that I've done. And so now, yeah, I kind of wonder if, in a way, having the protein source as egg white in combination with the omega-3s was affecting some of the results. Yeah. Yeah.
Other questions? Oh, maybe. Oh, sorry. Yeah, go ahead. Go ahead.
Thank you very much for the information on the mixed effects of, the good effect of mixing the proteins, plant and animal. That opened my mind. I didn't know that was happening. And could you extend more about why is that happening? Yeah, like that slide on
Yeah,
Richard Believer.
Yeah, and this one.
So to understand, you're saying that plant proteins are better to decrease the fat in the liver?
Well, there's examples of plant protein only, right here, soy protein, egg white protein only, animal protein, and combinations of them. And so I'll say the bottom line is I don't know exactly what's causing this, right? Usually with proteins, the first thing we think about, well, what's the amino acid composition? And so we do have the amino acid composition done on the different protein sources we've done, and I've kind of looked at it. And I don't see anything there that explains it. Right, there's some literature around particular amino acids like leucine, isoleucine, and so forth, but I don't think the differences in these diets explain that, right? So, so we can go, okay, is it amino acids? Is it something about protein structure? Is it something around digestibility and absorption? I don't know. In our second study, we did collect samples so we can do a gut microbiome analysis. So we are kind of looking at that point because in the protein field, there's interest in that. So we'll take a look at that. Are there particular gut receptors that say are being affected by different compositions of protein or something that then are impacting metabolism, and that's what's going on in the liver. So it's kind of like, I think I hope some other people follow up on this, but yeah, it's kind of interesting, and we didn't expect this either.
Thank you.
Yeah.
Thank you very much for the lecture. I don't know anything about this, so the question is about omega-3. There are lots of omega-3 supplements on the shelves. So is it better or is it enough to get them from the bottles, or is it better to consume salmon, for example, or other food which contain which has this kind of fatty acids?
Time, right, and great question, and I think probably nutritionists are still discussing, right, kind of your particular question, but I would say, one, based on what I know, I think it is important to get more omega-3 in our diet, right? So by eating more flaxseed, right, that can help. Then there's questions about, is it better to more directly consume EPA and DHA? And certainly when we do our studies, we can see advantages of that. And then your question is, right, do we get it from food, right? So from fish or supplements, right? And again, both of those work, right? I mean, our human studies we do with supplements because that's the easiest to manage in a study. And we can show those work, those are effective. But on the other hand, if we're consuming like a whole food, right, like fish, salmon, then maybe there's some other additional advantages there, right? And then the other thing that comes into play too, right, is cost. Because when you live here in Winnipeg, having ocean fish, having salmon, right, is costly. So, so there's those different factors about how to balance it, right? When we consume fish or fish oil capsules, we're getting the combination of EPA and DHA, right? And so that's a good thing. I mean, there's research around, what the balance of, which one versus the other, but when it comes down to our kind of just our practical life, just getting more EPA and DHA is good, okay? This reminds me, I think I brought, I just want to look at my,
Can I ask on that, Carla?
Yeah.
Is there any difference between farm salmon or wild salmon when you're looking at those levels? Are you getting an equal amount, or do they have to be deep ocean?
see, well, so that's why, like, there's measurements being done, and you can kind of look it up. Like with the farm salmon, it really depends on how well they're fed in terms of the source, right, to get the omega-3 level there, right? So there's that aspect to it. With the deep ocean fish, right, it's the cold water temperature which is causing the production of more omega-3, so those fish can, say, supple in the water. And that. So, but yeah, but there are papers published on, you know, comparing different sources of fish in that, and even looking at, you know, say in northern Manitoba, from, you know, colder water lakes. I mean, yeah, there's a little bit more omega-3 there than there is in fish that are from warmer waters or whatever. Right. And I guess along this line too, it's also thinking about like omega-3 eggs, for example, right? That's another avenue of how getting more omega-3 in the diet. This omega-3 index is what we're doing in our study. And so it's taking red blood cells, and it does a kind of a calculation of how much EPA and DHA you have. And because it's red blood cells, and they're there for a longer period of time, it kind of reflects your status over a longer period of time. And so this graphic, and there's many more like them, is looking at this omega-3 index and risk for heart disease. And when you see something like this plotted across different countries, it's like Japan population is kind of over here, and North America is kind of more over here. And so what we're finding right now is people in Manitoba who have come into our study, omega-3 index is kind of typically between about 4 and 6. We've got a handful of people kind of over here. We've got one person who's actually at 12%, and I really like to find out more what they're doing to get to that level. But you know, just in general, most of us could kind of be here, and based on literature and studies that have been done, yes, the recommendation about eating fish, high omega-3 fish a couple times a week is what you need to push over here, or if you're taking supplements or just having a good diet with other plant-based sources of omega-3. So.
Perfect. Thank you for the questions. I better stop us there. Let's thank Dr. Taylor one more time, and thanks for being here.